I will discuss the functions of a cell membrane. According to the cell theory, cells are the main unit of organization in biology. Whether you are a single or a blue whale with trillions of cells, you are still made of cells. All human and animal cells are contained by a cell membrane that keeps the pieces inside. When you think of a membrane, imagine it is like a big plastic bag with some tiny holes. That bag holds all the cell pieces and fluids inside the cell and keeps any nasty things outside the cell. The holes are there to let some things move in and out of the cell. The cell membrane is not a solid structure. It is made of millions of smaller molecules that create a flexible and porous container. Proteins and phospholipids make up most of the cell membrane structure. As we were talking about the bag earlier, if the bag ripped, then all the bad things would go in the bag and all the good things would go out, which would make the cell die because it needs all the good things to keep it alive. Red blood cells are highly abundant, circulating in the vertebrates, the major function of which is respiratory gas exchange. However, other functions, including an interaction with the immune system, have been attributed to these cells. Most of your blood is made of red blood cells. The red color comes from a pigment in these cells called hemoglobin. Red blood cells transport oxygen from the lungs to body cells where it is used to make energy during respiration. Oxygen combines with hemoglobin to form oxhemoglobin. Guard cells surround each stoma. They help to regulate the rate of transpiration by opening and closing the stomata. I have chosen the red blood cell and the guard cell. As you can see, the red blood cell has only two things inside of it: a cell membrane and a cytoplasm. In the guard cell, instead of the cell membrane, there is a cell wall and no cytoplasm. The only thing in common is that they are both cells. I am going to be talking about Fiona Wood. She is a British-born plastic surgeon working in Perth, Western Australia. She is the director of the Royal Perth Hospital Burns Unit and the Western Australia Burns Service. One of Fiona's early achievements was the development of skin culture lab that she co-founded with scientist Marnie Stoner. Professor Wood and Marnie recognized the potential of tissue engineering technology to treat burns, and in 1993 developed a skin culture facility with support from a telethon grant. Their product evolved from confluent sheets of CEA to aerosol-delivered cell clusters and is known as spray on skin. This technology, commercialized through clinical cell culture, is the world's first and has been used on more than 1,000 patients around the world. In 2005, Professor Wood and Marnie won the Cluny's Ross Award.